Okay, so we're going to move on to the last of the two major macromolecules of life, and that is going to include the nucleic acids. All right, so the nucleic acids, like proteins, have some complexity to them. All right, and nucleic acids are really composed of three major components to them, if you will. Okay, the first thing, these nucleic acids contain a sugar, okay? They contain a carbohydrate. And they contain a 5-carbon sugar. It's a sugar that has 5 carbon atoms. Okay, so a 5-carbon sugar, all right? And that is known as, let me see if I can find a better, darker one here. That is known as pentose, right? Like pentagon or pentagram, 5 sides, so 5 carbon. Sugars are known as pentoses. All right. Is anyone taking microbiology here? All right, I'm putting you on the spot. What pathway do these pentoses come from?
We never went into that.
What? You didn't go over metabolic pathways in microbiology? Oh my goodness. Okay. It's the pentose phosphate shunt or the hexose monophosphate shunt. You don't have to specifically know that for this class, but yeah, pentose sugars. Okay, so you have 2 flavors that are important in nucleic acids. Okay, you have what's called ribose. Okay, and this is the sugar backbone in DNA, or excuse me, RNA. RNA. Okay, and then you have deoxyribose. What do you suppose deoxyribose is? Yeah, DNA. And specifically, it is a ribose with 1 less oxygen. Deoxy. Does that make sense? Right? Right. So that's a difference. Okay, but in both cases, both DNA and RNA, you have a 5-carbon sugar. That sugar is attached to a phosphorus or phosphate group. All right, so you got phosphate. And then finally, that phosphate group is attached to a nitrogenous, what is known as a nitrogenous base. All right, nitrogenous base. And so, just to super simplify it, okay, you're going to have something that kind of looks like this. Okay, phosphorus, and then the phosphorus is going to be bound to your 5-carbon sugar, okay. Oxygen coming off the end of it there. All right, and then that is actually what binds to your nitrogenous base. So you have your pentose here. Okay. You have your phosphate group here, and then you have your base here. Okay, and these bases, okay, so this is one side of your DNA. In DNA, when talking about DNA specifically, does it have 1 or 2 strands? It has 2 strands, right? So this is one strand. And the 2 strands are actually form what's known as an alpha helix, right? Okay, so they have that. So we're just looking at one strand. Okay, so we're unraveling it and looking at one strand, and then I have another strand over here. These bases often have hydroxyl groups on them. Uh-oh. What happens when a hydrogen is covalently bound to an oxygen? Is that a polar and nonpolar situation? Polar, right? The oxygen pulls electron density away from the hydrogen. So then what I have is I have a similar kind of situation over here on this side. And again, I'm kind of super simplifying it, but that's okay. All right, so I have something like this. Okay, like that. On this side, this is my base. All right. And then that base is attached to that 5-carbon sugar. But the orientation of that 5-carbon sugar is oriented in a slightly different direction. See how the tip is kind of oriented up like this, up in this direction here, and it's kind of down in this direction here. That'll be important in just a minute here. And then I have the phosphate group here. And what do you suppose holds the two ends together, these two bases here? What kind of force do you suppose is going on here? Oh, our good old friends, a hydrogen bonding, yes. You have hydrogen bonding between the base pairs. So you have hydrogen bonding, holds the base pairs. The base pairs are attached to a pentose, which is attached to a phosphorus, or a phosphate group. Okay, you guys okay with that? And so what I'm going to do now is I'm going to erase this, and as good as I try to get my art, it's just not going to be great. Although I have improved substantially. I really have. A few years ago, it was all stick figures, so there's been some substantial improvement. I'm just going to turn a projector on. I actually have a picture that I'm going to project here where it's a little better, and it actually has several of these base pairs. Now this here, this phosphate, pentose, and a base by itself is what we call a nucleotide. Okay, that is a nucleotide. All right, nucleotide. Okay, I'm just going to turn this on. I'm actually going to leave the screen up so I can kind of draw on the board on top of the picture, hopefully. Yeah, all right, that's pretty good. Let's do this here. Get more of it on the board if I can. Okay, whatever, that'll work. All right, so let me turn the lights off here to show you guys a few things. Hey, all right, so I've got my, you guys see that okay? My phosphate, my 5-carbon sugar, and my nitrogenous base. You guys good with that? So I have kind of the backbone of this. The backbone is my phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. You guys see that? That forms kind of the backbone. Same thing here. Phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, etc. You guys cool with that? But the sugars are oriented. On this side here, you see how the sugars are oriented kind of like this? And I'll just draw an arrow. The oxygen on that sugar is oriented in this direction here. You guys see that okay? And then over here, the oxygen is oriented in that direction. You guys cool with that? So the carbons are also oriented a little differently. And so what that does is, that kind of creates where this attachment is on the sugar, where this is attached to the carbon. Okay, right here, this is on the 3rd carbon of that sugar. So this is what we call 3'. Okay, that's, if you ever see that, that's a 3'. Okay.
How do we know that it's the 3rd carbon on that? Where are you counting from?
So what we do is we start, we basically start here 1, 2, 3, 4, 5. So we start next to this oxygen here. See this oxygen? Mm-hmm. And then there's a carbon here. Okay, so that's where we start. So it's somewhat arbitrary, but everybody's agreed. Okay, that's where we'll start. Over here, it's the same thing. It starts here, or over here rather by the oxygen. 1, 2, 3, 4, 5. Okay, so you can see that the oxygen is attached to the 5th carbon here and the 3rd carbon here. Likewise, up here, it's attached to the 5th carbon, whereas it's attached to the 3rd carbon here. So what does that mean? Well, that means that this side here, going from up to down, runs in a 5' to 3', okay, where they're attaching to the sugar on the carbon. And then on this side, okay, it goes from 5 down here up to 3, okay? And that's just where it's attaching, just the carbon that it attaches to. I'm not going to ask you any questions about identifying where the carbon is that it's attached to, okay? So don't spend all day trying, oh my gosh, how do I count the carbons? That's not the point. The point is that on one strand, it runs in a 5 to 3 going down, and then on the other strand, it runs from 5 to 3 going up. Are you guys okay with that?
Does that create its shape at all?
Well, it does help in creating, as this coils up, yeah, this helical, this alpha helix here.
It seems like they follow each other.
Yes, yeah, they do, they do.
Yeah.
And then you imagine that this gets wrapped up. Okay. So again, this is a nucleotide. This is a nucleotide, okay? And then they are chemically bound by hydrogen bonds. Now how this works is there are several different nucleotides. There are 4 of them in DNA, okay? There are 4 different base pairs, rather, okay? Base pairs on each of the nucleotides. So let me turn this off. So with DNA, okay, so we'll start at DNA, okay? We have adenine, okay, thymine, guanine, and cytosine. And the reason that I drew them like this is adenine can bond to thymine via hydrogen bonding, and guanine to cytosine. So ATGC is the way I think of it. Okay? So only adenine can bond to thymine, only guanine can bind to cytosine. You guys okay with that? That is in DNA. Now in RNA, there is a slight modification. Okay? There's a slight modification to RNA. Let me just do this in a different structure. What happens is thymine is replaced by something called uracil. It's very similar to thymine. Okay, so adenine, okay, can bind to uracil. Make sure I spell that right. And then you have guanine binding to cytosine. Here. Okay, cool. So how does this work? Well, what we found is that a certain sequence of base pairing, and when I say base pairing, I mean adenine, thymine, a certain sequence, so let's say that I have A, G, T, C on this end, on this strand of my DNA. Okay, what will I have on the other end here? T, T, C, A, G. Okay. And what we found is that, let's just say, I don't know if this is the case, but let's just say that this particular arrangement of base pairs, okay, when turned into RNA and fed into a ribosome, this sequence of base pairs tells the ribosome how to make blue eyes. Okay? This is the code for blue eyes. Okay? This chunk, this section of base pairs, is known as a, I'll see if you guys get it. It's a sequence of base pairs that codes for a certain trait, a certain protein.
Gene?
A gene. Yes. This is what's known as a gene. All right? This is what's known as a gene. You guys okay with that? Now, I'm not saying that this is literally the gene for blue eyes. I don't know what the gene is, but it is some sequence of base pairs. Okay, that is a gene. Okay? Yes?
How do genes end and begin?
What do you mean, how do they end and begin?
Is there any point where they break? It's like blue eyes, okay, now here's brown hair, or are they all different strands of
Yeah, and
acid
I don't.
entirely?
Yeah, and I don't know exactly, I don't know where the gene for blue eyes is in the genome.
From your parents.
And what's that?
They come from your parents, depending on your parents' cells and how they
Yes,
combine.
yes. You get, and when we talk about sexual reproduction, yeah, you get half of your genetic material from mom, half from dad, and it kind of gets mixed up in your genome. But to say which chromosome it's on, I don't know which chromosome. I do know there are certain diseases, genetic diseases, and there are two general types of chromosomes. You have what are called your, we'll start at the basic, your sex chromosome. Those are your X's and your Y's, okay? And then you have all the other chromosomes which aren't sex chromosomes, and those are what we call autosomes. So if somebody has an autosomal disease, that is a defective gene on one of the other chromosomes that is not a sex chromosome. And if they have a sex-linked disease, then that is involving a gene on one of their sex chromosomes. And then you can even add another level of complexity, what we call a recessive and a dominant gene. And a recessive genetic disorder, okay? Recessive means that you need to get both the gene from both your mom and your dad, okay? Both your mom and your dad has to give you that gene. Otherwise, if you only get one gene, then the other side of the DNA can be copied and your body's okay. You're like, oh, okay, I won't do that. But a dominant disorder, okay, a dominant disorder is if you only have one, doesn't matter. If you just have one gene, you'll get the disease, okay? And then you can have what's called an autosomal recessive disorder. So that's a recessive genetic disorder of an autosome chromosome. And then you can have an autosomal dominant disorder, which is a genetic disorder that's dominant involving one of the autosomes, so on and so forth. We can even identify the specific chromosome where the defect is. And some defects involve extra chromosomes. There is one called Trisomy 21 involving the 21st chromosome. Does anyone know what Trisomy 21 is? Down syndrome. There you go. So yeah, does that kind of make a little bit of sense though? Hopefully. But yeah, I'm sorry. I couldn't tell you I'm not sure where blue was it. Is it the 20th chromosome I don't know exactly where. But I will tell you a little bit about something known as a telomere. Okay, have you guys ever heard of telomeres or telomeres? So telomeres are kind of caps. Think of them as caps, okay, on your DNA. And when your cells divide and copy themselves, which is something we've talked about in lab, okay, when they do that, we know that it's not always perfect, right? It's 99+% perfect, but there are always little imperfections and little things that can happen. And one of the things is you have this, kind of like this junk DNA, if you will, that caps on the chromosomes. And basically what it is a buffer zone. It's a buffer zone that can kind of take into account these little errors that happen. But what happens is each time a cell divides, those telomeres become smaller and smaller and smaller and smaller until eventually you run out of it, and then the cell can no longer compensate for these little errors, and then you end up having problems. And this is one of the mechanisms behind aging. As we age, as our cells divide, our telomeres become smaller and smaller until we end up not able to really efficiently divide. And what happens? Well, the skin becomes wrinkled, your organs begin shutting down, you become more prone to problems. That is one of the many mechanisms behind aging. But that's just one that I figured I'd throw out there. Okay, so these genes are wrapped up in DNA, and then the DNA itself gets wrapped around little protein balls called histones, right? And then those get wrapped up into the chromosome. Is that, you guys okay with that? Okay. So far so good? Make sense? So going back to the central dogma of molecular biology, you have all this DNA wrapped up inside the nucleus, all protected. Okay. What happens is the DNA gets copied. We make a copy of it. So let's say that I need to make some protein, right? Instead of literally taking the actual DNA out of the nucleus and potentially exposing it to becoming damaged or lost, we make a copy of it, and that's called transcription. Okay, so the DNA in that area, okay, in that area of the chromosome gets pulled out, and there are proteins, there are enzymes that help unzip this. One of them is called DNA helicase. Okay, it helps open it up, zip it. It gets copied into RNA. The RNA then leaves the nucleus, goes through the nuclear pore, and it takes a ride on the sarcoplasmic reticulum, okay, or the endoplasmic reticulum, excuse me, until it runs into a ribosome. And then it gets fed into the ribosome, and it tells the ribosome which amino acids need to be combined to make that protein, be it a channel or something else, a receptor. Does that kind of make sense? Mm-hmm. All right. That's the general picture. There's a little nuance there. We have special types of RNA. Okay, we have a type of RNA known as rRNA, and that is RNA that exists where in the cell?
Ribosome?
Not the ribosome. Well, it has something to do with ribosome, but it doesn't exist in the ribosome. That type of RNA exists in the nucleolus. So remember in lab we talked about the nucleus, and then the nuclear envelope, and then in the very center of the nucleus you have the nucleolus. Now the chromosomes are not in the nucleolus, they are around it, and in the nucleolus is where I have a special type of RNA called rRNA, which stands for ribosomal RNA. Okay, so this is when our body needs to produce, when the cells need to produce ribosomes. Okay, that's where that RNA comes from. You guys okay with that? Okay, it does not come from the surrounding stuff.
But you said it's called ribosome.
rRNA or ribosomal RNA. The RNA, the run-of-the-mill RNA that comes from DNA is what's known as mRNA or messenger RNA. Okay, so you've got mRNA, rRNA. Okay, and you actually have some other types of RNA, but those are the two big ones I want you guys to focus in on for today. All right, you guys okay with that? And there is another area in the body that contains its own genetic material, an organelle within the cell that contains its own genetic material. And this is just crazy to me to think about what we found out about this. What organelle contains its own genetic material? And this is a question that you guys had to answer in lab as well. Say it? The mitochondria. The mitochondria contain their own genetic material. This is how crazy this is. What we found through gene sequencing, and what we found is that the mitochondria is not a classic organelle. What it looks like is that millions, at least hundreds of millions of years ago, perhaps longer than that, mitochondria were actually bacteria that got absorbed by a cell. And instead of killing the cell, or instead of killing the bacteria, a symbiotic relationship developed where this bacteria was able to produce energy for the cell and so the cell kept the bacteria around, and then over millions and millions and millions of years, that has just evolved into all animal cells having these mitochondria. But all the evidence points to these mitochondria actually being probably a bacteria that got absorbed into a cell. And that's why they have their own genetic material. Because at one point in time, in the very distant past, they were their own organism. How crazy is that? And we can actually sequence that genetic material, and that is very important when it comes to lineage, right? And particularly when it comes to mom. Okay? The genetic material of mitochondria follows the mom. So we can follow our maternal lineage back. And some of you guys have heard of the term Mitochondrial Eve. Has anyone ever heard of that term, Mitochondrial Eve? And that's basically where we've tracked the mitochondria back through generations and generations of human beings back to more or less the first human-like female, and we've been able to see when you look at it, we've been able to see how human beings have migrated out of Africa and how they've gone by tracking this genetic material in the mitochondria. Really interesting stuff. That also means that there can be mutations of that genetic material, and you can have certain diseases. And I believe that you guys had to answer questions about one of these diseases called a mitochondrial cytopathy. Does that ring a bell? Anybody? Yeah, it was in the check your understanding, the test your understanding in the lab. So there are various disorders of the mitochondria, okay, where you have mutations of those genes and they don't code for the proteins that those mitochondria need. And those mitochondria are not as good or as efficient at producing the ATP that your cells need, the energy. And which cells do you think really need lots of mitochondria? Which cells in particular? Muscle cells, right? Muscle cells. Because your muscles are always moving, always working, using lots of ATP. So some of these mitochondrial cytopathies can involve, people getting very weak and having musculoskeletal problems, or their muscles aren't working, and maybe they have problems breathing, and have problems with metabolism. Okay. You guys okay with that? Does that kind of ring a bell a little bit now?
Yes.
And I know several people kind of had some problems with that, so you'll see that I wrote notes on some of you. As long as you tried to answer the question, you got points. So if you tried to answer, you got points. If you just didn't bother to answer, I didn't bother to give you points. But if you tried to answer, you got points, and then I just wrote some additional information. So definitely go back through your labs and look at the comments that I wrote, because that'll help. Okay. So there we go. Let's see. Before I put you on another break, let me give you an example of another. So that's a mitochondrial disorder. Another relatively common genetic disorder. Anybody here looking at respiratory therapy? I'm trying to cover all the I'm trying to focus on every discipline. So there is a. Oh, say it. Say it.
What?
Oh, did you say it?
Cystic fibrosis.
There you go. Cystic fibrosis. Yeah, cystic fibrosis is one of the classic genetic disorders that we talk about, involving defective genes for a certain type of ion channel, chloride channels specifically, where we can't move chloride and water in and out of cells like we should, and that produces mucus that is very thick and sticky. And it builds up, and you can't get rid of that thick, sticky mucus, and it tends to cause infections and lung disease. So that's yet another example of how these mutations can work. Okay, so before I put you guys on break, I just want to briefly talk about this, because this will segue real nicely into cytology, and this will segue real nicely into the second most common cause of death. Which is what? Cancer. Yeah. Second most common cause of death, okay, in the United States, cancer, right? There's a significant chance that you guys are all going to get cancer in your lifetime. Very significant chance that you're going to get some form of cancer. So it's a big deal. It's one of the big health problems that we deal with. Some cancers we have really good treatments for, others not so good treatments. But what we found is that in the human body there are certain genes that prevent cancer from developing. These are known as tumor suppressor genes. Okay? And depending on environmental conditions and toxins and things like that, these genes can be turned on or turned off. And in a lot of cases, what these genes do is these genes allow cells to identify problems. And generally, when a cell identifies that there's a problem, oh, I'm not functioning right, I'm not doing my job right, the cell will kill itself. Okay, the cell will kill itself, will deactivate itself. And that is what's called programmed cell death. And the term for that is apoptosis. So if a cell starts dividing in a way that's not good, okay, because that's all cancer is, right? Cancer is just cell division.
Defective cells.
Yeah, cells lose their ability to do their job and they start dividing uncontrollably. Okay? But if you have tumor suppressor genes that are working, you've got a good immune system that's working, there will be a response, okay, that will tell these cells, hey, you guys go kill yourselves, and the cells will work. Right?
So how often
Yeah.
does our body produce defective or cancerous
We
cells?
think that you probably develop cancer several times in your life. Every human being develops cancer, and for the most, generally what happens in people that don't go on to develop systemic cancer is the immune system responds, and all of these built-in mechanisms, okay, that tell the cell to kill itself, it listens and they kill themselves and you get rid of it. Yeah, but then there are cases where it doesn't happen, and obviously we're still trying to figure all this out. But yeah, so that's apoptosis. It's programmed cell death. The cell kills itself to spare the organism. Now, when cells die due to damage, like direct damage or severe hypoxia, and they just die off, that's something called necrosis. Now, there are also other genes that, by themselves, they're not particularly active. They don't do anything. Okay, by themselves, they're not doing anything, and they're what are called proto-oncogenes. They're called proto-oncogenes. Proto-oncogenes. All right. And what happens is these genes can get turned on, okay? And when these genes get turned on and they start activating and they start producing proteins, okay, they become oncogenes. And these are genes that can produce cancer. Okay, so this is yet another mechanism for cancer. Is a proto-oncogene gets activated, it gets switched on through a variety of things can cause them to switch on. It gets switched on, and then it starts coding proteins, and then that leads to this whole a whole chain of events that can eventually lead to the development of a cancer. Okay, so cancer is very intimately connected to DNA, very intimately connected to genes, is very intimately connected to mutations, okay? Mutating these genes, mutating the DNA, and cells copying genes that tell them to do things that are harmful, that ultimately tell the cells to divide uncontrollably and to lose their specialized function. That's classically what we find with cancer cells, is that they divide uncontrollably and they do not function like they should. Okay, so they quit their job and then they divide uncontrollably and they cause problems. And with that, I'll see you guys in about, let's take 5 minutes.